Hey everyone, I'm Mariah Mitchell. I apologize I can't be there presenting this to you in person, but I still want to share my project, which was sorting double haploids using a Qsorter. So just a little about me. I go to Iowa State University. I'm getting my master's in seed technology and business, and I had the privilege of being a corn breeding research intern at Marshalltown this summer. And so these pictures are me running the Qsorter, which we'll get into, and me doing some planting. So the QualiSense Qsorter Explorer, it's similar to your typical color sorter that we see in processing and cleaning seed, but this one only looks at the embryo. So it's recognizing kernels with a purple embryo and a colorless embryo. And so the Qsorter is for separating haploids from diploids and outcrosses to be used in the double haploid process. I know that's a mouthful, but what you need to know is that it's sorting out what has a purple embryo and discarding it, which would be diploids, and it's keeping the colorless embryos, which are hopefully haploids. Just some basic terminology before we get started. What is a haploid? A cell or an organism with half the number of chromosomes. And so in humans, just like corn, we are diploids. We get half of our chromosomes from our dad, half of them from our mom, and with a haploid, you're only having half the set of chromosomes. So in this case, with our double haploid process with corn, they only have the female chromosomes, no male. A double haploid is that same female plant with the female chromosomes, but doubled. So two sets, but both being from female rather than one from mom, one from dad. Why double haploids? They are a way of creating completely homozygous inbreds in a reduced amount of time. So typically inbreeding takes around 7 seasons of selfing the same plant over and over again till we have pretty much solidified the genes we will use to breed hybrids. With double haploids, we're creating an inbred in about 3 seasons. We're taking out the selfing process by just using the genes that we want and doubling them, adding no more. Just a little bit more on the double haploid process. The desired female is crossed with a male called the inducer. So that male inducer contains a mutation that is carried to the female when it pollinates it that spontaneously deletes all of the male chromosomes. So what's left is a female with half of the chromosomes, none of the male DNA, making it a haploid. Those haploid kernels are introduced to a chemical that doubles those chromosomes, creating a double haploid, just as we had mentioned before. And so now we have a female plant that is completely inbred, containing no male DNA. In the double haploid process that first summer, females and male inducers are planted in a field with a 2-2 pattern. As you can see in the right-hand corner, you can see the 2 female rows planted right next to the 2 male inducer rows. Females are also detasseled because all we want is that pollen from the inducer. Males will pollinate the females, creating that mutation, deleting their own chromosomes. The female plants are harvested, and that seed will be sorted from diploids, haploids, and outcrosses using our Qsorter, or what is typically done is that we'll send it to Chile and they'll sort it by hand. That same company that sorts the haploids also doubles them with colchicine. Colchicine is that chemical that's a genome doubling chemical, and so they'll put it in the media, and those haploids will double, making them double haploids. What we get back from those are completely usable double haploids, but we like to increase them. We'll weed them out for any bad seed, unwanted mutations, and strengthen the plant for breathing. So we'll plant them again ourselves. So from that process, you'll have a homozygous inbred that will be used for hybrid breeding. The main use for the color sorter is based on this linked color gene. So the inducer plant has a color gene linked to it to help us identify the difference between haploids, diploids, and outcrosses. So that purple gene will show up in the embryo and the endosperm of the kernel, and it'll also show up in the root of the germinated plant. So in the lower left picture, you can see if you were to just take a handful of what was harvested out of the field, you've got colorless embryos, which are outcrosses. You can see purple embryos, and you can see purple on the endosperm as well. On the right, I germinated some diploids just so we could see kind of what they looked like root-wise, and you can see it also showed up in the shoots a little. I'm a visual learner, so I made these pictures to help us better understand the color gene and kind of how it gets its color based on the fertilization of corn. So corn is double fertilized, meaning 2 sperm cells, one enters the embryo and one enters the endosperm. So in the left-hand picture, you can see pollen will land on the silks. Those sperm cells, one will end up in the embryo, the blue ones, and one will end up in the endosperm. If we move to the right, looking at the diploids, let's say we have our inducer that has the purple pollen gene. That purple pollen gene will end up in the embryo and in the endosperm. And let's say the doubling didn't happen, it just didn't work, so both are purple. We don't want that. We want to make sure male chromosomes are deleted in the embryo, making it a haploid, and visually diploids will have a purple endosperm, meaning that the male DNA is still there. Moving over to the haploids, only the cap of the corn, the endosperm, will be purple. That's showing that the male genes, which would be purple, were deleted in the embryo. Over to the outcrosses, there'd be no purple at all because the inducer had nothing to do with pollinating or fertilizing. These are some pictures I took from corn that I was running through the Qsorter. As we can see, outcrosses have no color. Haploids just have that purple in the endosperm because the male DNA was deleted in the embryo. And diploids, on the other hand, the deletion did not happen, and there's purple still in the embryo as well as the endosperm. Here is a short video of the Qsorter in the works. So here is the sorter explorer. It's missing the X. This is the inside of the front, the back. And so when you go to get her started, you'll press start and reset. You'll open the application on the computer, operator, sorting, and this is where you get to choose your kernel size and your belt speed. After running a few tests, I found that it picked up the kernels consistently at the large size and the 30 kernels per second speed. You'll name your batch, you'll fill the hopper, and you'll press start. It'll do its checks. And it'll start to pick up seed. Right away you can see it's already sorting the seed. Bin 2 is the diploids, bin 3 haploids, and then bin 1 is unclassified. So those are the seed that the machine couldn't quite identify the first time, so it reruns it. In the lower right hand corner you'll see the number 28. That is the objects per second, now 27. It's moving around 27 kernels per second. When it's set for 30, that's pretty good. Bin 3 is the haploids, bin 2 is the diploids. And so this is a close-up of the camera taking pictures in the belt, moving those seeds along. So it's pretty interesting to see how fast it can snap those pictures. This is diploids versus haploids. And then once you are at the end, you'll rerun the unclassified to get your percentages, and you can go into the summary of reports and you can get any information that you're looking for, mean, standard deviations, percentages of sorting, really anything you can think of, and you compare them to other summary of reports as well if you needed. There are some faults to the double haploid process, that being only 8 to 12% of the seed that we'll take from the field are putative haploids, meaning after the cross happens and we harvest that seed, only 8 to 12% look like they're haploids. Of that 8 to 12%, only 8 to 12% of that will survive the doubling process. So you can have 40,000 kernels that are harvested and sorted with the Qsorter. 4,000 of those will end up being haploids. 400 of those will end up surviving the doubling process through the colchicine. So you'll only end up with 400 kernels, but that's still a lot in terms of how much time you're saving and as well as money. The sorter only looks at the embryo. As we had mentioned, outcrosses have no color just as well as haploids, so outcrosses will be sorted into the haploid bin. This isn't necessarily a huge issue because you can tell a haploid from an outcross because of the purple endosperm. The color gene might also get lost or unlinked to the inducer. That means if the inducer loses that color gene, we won't know if the female was pollinated or fertilized by the inducer and so forth. It's just important to manage your inducer in that sense. Some pros of the double haploid process and the Qsorter. Despite the low quantity of seed that you'll collect after the process is over, it's so worth it. The time of 3 seasons versus 7 and the money spent of the typical inbreeding process is so crucial when you're trying to move your product down the pipeline towards more testing. Money from large shipping quantities and services are saved by doing the sorting at the location of harvest, doing the sorting at Marshalltown after we had harvested our own seed. We don't have to send the large bag of the mixed diploids, haploids, and outcrosses. We can just sort them ourselves, take what's left in that haploid bin, and send that over so they can double it. We don't have to pay for them to sort it themselves. We don't have to wait for them to do it. All we're doing is the doubling there, and we'll get it back in time for planting. It's easy for this sorting to be done at the end of the shelling line while it's already being processed on our own. And that is it. I want to thank you guys so much, Bex, for giving me the opportunity and the independence to work on this project myself. This is by far my favorite internship, and I'm so glad I had the opportunity to work for a company that has the same values as me. This is a lovely picture of the 3 other interns I got to work with as well, so I'm glad and appreciative of the opportunity to have met them as well. So thank you guys.